Hello and welcome to this special edition of On Cue Insight. In the next half hour, we are going to learn just a tiny bit about what it's like to work for one of our region's most prestigious and important facilities, the Hormel Institute in Austin. Dr. Ted Hinchcliffe joined the Hormel Institute in 2008 and is an associate professor and section head there. Prior to coming to Austin, Dr. Hinchcliffe taught for 10 years at the Marine Biological Laboratory in Woods Hole, Massachusetts. Welcome. Thanks for coming in.
Thanks. It's good to see you.
And we're going to learn a lot about cancer research and what the Institute's all about. But first, let's back up a little bit. Your education growing up was science and, research, microscopes. Was that a big thing for you even as a youngster? Maybe there's young people watching wondering how you get to be a professor.
Absolutely. My education growing up, I went to Montessori school. There was a lot of exploration that was involved, a lot of nature camps. This was during the 1970s.
In Ohio?
In
right?
Ohio.
Okay.
I grew up in Dayton, Ohio. Then I went to college there and studied chemistry, and wasn't really sure what I wanted to do with myself, and had done a research experience while I was an undergraduate, and that really got me involved in deciding I'm going to go to grad school and pursue this career.
Mm-hmm. And so it was, biology in high school and college, or just general studies? What?
I took a lot of advanced science courses. The Dayton Public Schools had a fantastic career academy which offered advanced physics, chemistry, biology. I had probably taken more chemistry and physics than biology at that time, and I was a chemistry major in college. So I was taking biology courses, but I was really taking sort of a variety of things.
So even in high school, it was
Absolutely.
Strong on science as a real passion of yours, because this is kind of, in the few times I've been there, it's a calling kind of thing, because you're a university professor also. There are much simpler ways to have tenure and then work at an institute like this. There are a lot of extra duties.
Yeah, it's a very exciting environment, and I came here from Notre Dame, so a very traditional university environment, being in a biology department, teaching and doing research, and now I'm doing just research. I spend a lot of my time writing research grants, writing papers, but I also get to work in the laboratory, and I spend a lot of my time actually working at the bench, and particularly at my microscopes.
And we should point out, for truth in labeling, we're both Austin Packer basketball fans, because that's really where I got to know you.
Yeah, and that's been really fun. My wife played basketball in high school and in college, and really got me involved in watching high school basketball through her father. When we moved to town, that's one of the things we started doing, and that's where we met you and got to talk about watching basketball, which is great. So
the Institute, you are one section leader of about 10 or so, and there's other researchers. Run down the org there.
Yeah, so we have 10 to 12 section heads, which is sort of in flux. People are coming in and people are moving on. But that's really changing now because of the expansion. So now we're going to start moving up into the realm of about 25. That gets us to the size of a really standard academic department. And that allows us to do a lot of things that you can do. So you have those 25 section heads, each person runs a lab, then they have people working with them, postdocs and things.
So you're a section head, so Dr. Hinchcliffe has this concept or specialty, and then you have folks beneath that are helping you try to answer or unlock questions that you're having grants to solve.
Yes.
Sort of.
So postdoctoral fellows, who are people who have just gotten their PhD, it's essentially equivalent to a medical residency. So you work for 5 or 6 years in a laboratory, you learn how to run an independent project under a head like me. We have research technicians who do a lot of the work, and then we do a lot of the work ourselves.
And in doing the scant research I did preparing for this, I got to your bio page, and the first line of it had, I think, 5 words that I didn't know what they meant. And so I said, I'll just ask Ted when we get here. I know it has to do with cancer and cells and dividing in some way. Now, usually when you're sick, cells don't divide or they stop or somehow, but cancer, they keep dividing, or how many mistakes did I make there? Tell me a little bit about your work.
So we're made up of cells. We're made up of trillions of individual cells that are all stuck together that make up what our bodies are, all the organs and tissues that make up our body. And they start out as a single cell, a single fertilized egg that divides in 2 and 4 and 8, like the old TV commercial, the shampoo TV commercial where they doubled and doubled and doubled till they get to trillions. And our cells are constantly dividing even now. Some are dying off, some are dividing, and that helps us maintain our normal tissues. In cancer, something has become deranged in the controls that maintain that healthy balance between normal division, and now you get cells that start to divide abnormally. They go to the wrong place. They start forming into a mass of cells that's not where they're supposed to be. And at the heart of that is the division of these cells. So we're interested in how cells normally divide. What are all the different mechanisms that allow cells to divide properly? We want to know how that works so we can find out what goes wrong. So we can diagnose cancers very early on. We can be able to treat cancers in a very early stage. And ultimately, we'd like to be able to have the cure, something that can get those cells and get rid of those cells, and specifically those cells.
And so your scientific specialty is what, say it in the scientific world?
I work on mechanisms controlling mitosis, I work on mitotic spindle assembly, and I work on checkpoints that maintain genomic stability.
Okay, and mitosis
is cell division. Mitosis is the process of one cell dividing into 2.
Okay.
It's a very specialized time in the life cycle of an individual cell. There are massive changes in the organization inside the cell. The chromosomes, which are normally being used to make everything that you could possibly need for a cell to survive, suddenly become tightly wound up. So chromosomes are kind of like a skein of yarn, and they're unwound when the cell's using that skein of yarn, and then they're wound up into the skein when you're going to separate them. And so cell division is the process of taking chromosomes and separating them into 2 daughters. We're interested in every aspect of how that happens and what goes wrong when that process becomes maladjusted.
Thank you. And we'll be back. We're going to take a short break, and we'll learn more about what's happening at the Hormel Institute with Dr. Ted Hinchcliffe when we come right back.
You can watch On Cue anytime by visiting the KSMQ TV website, ksmq.org.
And welcome back to our special On Cue Insight edition. We're learning about cancer research, the important work being done at the Hormel Institute in Austin with Dr. Ted Hinchcliffe, one of the section leaders. And we've been talking about your research with cells. Does your work complement all the other folks there into some, or are they very separate types of work all leading to the same?
They're different types of work, and some of us intersect, and so we collaborate rather extensively. So I have collaborations with several of the researchers there, Zhigang Dong, who's our director, also Rick Brown, who works on some lipid transfer proteins, and Salim Bhatt, who works on pancreatic cancer. So being a small place, it's very easy for us where we have common areas of research where we can complement each other technique-wise. Conceptually, we do a lot of microscopy, so we help people out with that, and then we go to them to help us.
And I don't know if it's true with your work specifically, but I read when you have a discovery or something that there are other national groups that, like Dr. Bode collaborates with, so that when there's a big announcement, the Hormel Institute is part of it, but you're working with other agencies.
Absolutely. So I collaborate with a group still at Notre Dame from where I previously was. I collaborate with a group at Virginia Commonwealth University working on sepsis, an area that's completely different from my main research focus. But we're interested in the inflammation that leads to septic shock, which is a real problem with hospital patients. And so our collaborator in Virginia had come up with a neat idea. He collaborates with Rick Brown and myself, and we put together this group and have a grant from the National Institutes of Health to examine some of the molecules that are involved in and hopefully try to find a cure for sepsis.
And talk a little bit about that, the day-to-day work at the Institute, what it's like, what it looks like. Not for, I mean, you do a lot of work with grants, I'm sure, and that, but for the research piece, what you and your team, is it physically, is it sitting in front of microscopes, or is it mostly computers, or interviewing people? I mean, what are you doing?
So we don't really work with people at all. We work with cells, which we grow in little incubators in our lab. So
Which came from or a variety of sources?
The
or
ones we
mice,
have
or?
come from a monkey kidney tumor from the 1960s, if you can believe that. This was a cell line that was isolated by a group a long time ago and has been propagated. We get this from a tissue bank in Virginia.
Okay.
And we maintain these in the lab. So there's a lot of day-to-day busy work, making up chemical solutions, adding these to cells, allowing them to grow, doing preparations, and then we use those to conduct our experiments. We gather the data from those experiments, and then we sit in front of computers a lot, analyzing. Spend a lot of time using Microsoft Excel and Photoshop to put together all the pieces. We do a lot with microscopes, so we have some very advanced microscopes in my laboratory. Well, I think one of the things that drew me to this field was the very visual part of looking at cells, whether they're living cells that are undergoing the things they do, and we can watch that, or we've chemically fixed them, and now we can identify certain molecules, where they are, what they do, have they changed, have they moved, things like that.
And it's how those cells react to a mouse or something? Or when you say where we do our experiments, what?
So our experiments involve identifying particular molecules within those cells that may be either something that could go wrong in a cancer cell. So we've identified a particular molecule where there may be a mutation where that molecule doesn't function properly. So we can mimic sort of a disease state and then observe that in a cell and observe what happens to the cell. Does it behave normally? Does it behave abnormally? We're trying to find chemical pathways, biochemical pathways in cells that are involved in making sure the cell grows properly, divides properly. If we now do something to that, and we have a variety of ways where we can inhibit a certain thing from functioning, now that cell's not going to behave properly. And so we can very systematically take it apart and say, okay, if we change this piece, you're now going to have this result. If we change this piece, you're going to have this result. And then we can work our way back and say, okay, we think that's involved in a key process.
Would the before and after be a chemical analysis where you look at, here's what it was before and after, or is it a visual thing where you really see it? Wow, look how that changed.
So we can do both, and what we ultimately like to do is do that in a living cell. So we have a microscope that's set up with an incubator, so we can watch cells as they go through their life cycle. We can watch the events in real time, and then when we do our manipulations, we can say, okay, well, this chromosome should go here, but it doesn't. This cell should divide at this time, but it doesn't. This particular molecule should move from here to here, but we can prevent it, or we can make it go to a different place.
And this, impossible to generalize, but help me do this
Sure.
anyway, Ted. Is this happens in minutes or hours or days in the real time world?
It can happen on very vastly different timescales. We can have things that happen on seconds to minutes. We can have things that happen on hours, and we can have things on days. So we span the gamut of being able to observe those types of events. Some things we want to watch the life cycle of a cell, which is 24 or 48 hours. Some things we want to watch an event that may occur over an hour, hour and a half, and certain things where there's going to be a movement of a protein within the cell that can happen on the order of seconds. So we're equipped to be able to do all those types of things.
And I know the Institute has some giant supercomputers, but that's not really needed in this line of research?
For what I do, we're not really tapping into those supercomputers because we're not trying to do really computationally difficult experiments. Our computing, and we have some pretty powerful computing for doing our imaging, but we don't have to do the types of things that some of the people who will screen, this is a term, screening for a particular compound. the computational ability at the Institute is pretty phenomenal, given its size. We have 2 separate supercomputers, which are some of the fastest in the state, if not in the region.
That's really wonderful, and that's before this expansion project, which is, you mentioned it earlier, but it's basically going to just make room for more researchers.
Absolutely.
It's not more, it's for people in there, not machines or things.
We have a few labs to bring in some more sophisticated equipment and set that up. That's going to be dictated a lot by the people who we hire, and we're in the process of doing that. But most of those labs are, it's a research lab. It'll have a section head, people who work for that section head doing the same kind of thing. It just expands us out to a larger number.
We're talking with Dr. Ted Hinchcliffe from the Hormel Institute about his work fighting cancer, and we'll be back to continue the discussion right after this.
Queue up your own comments for On Cue and send them to us at onq@ksmq.org.
And welcome back to On Cue Insight. We're having a fascinating discussion about the work of a researcher at the Hormel Institute fighting cancer right here in Austin. From a large scale, Dr. Hinchcliffe, are we doing better with fighting cancer?
Absolutely.
Not just your work, but globally.
Globally, absolutely. The strides that are made are unbelievable, and it's difficult to be able to look back and see where we were 10 years ago or 15 years ago or 20 years ago. It happens as a very slow movement over time, but the way that we can address a lot of cancers today, we couldn't 10 years ago. We certainly couldn't do it 20 years ago.
Are people dying at the same rate that they used to?
They are dying at a slower rate depending on the cancer. There are certain cancers that continue to baffle us, but there are also cancers where people go in, they get a diagnosis, which is a really terrifying time, and then their physician tells them, this is one of the ones that we can treat. And so that happens more than it used to. And we continue to tick off these diseases where we now have, if it's not an outright cure that gets rid of that cancer, it's a manageable cancer. The problem is that we have some really intractable diseases, and those are continuing, and that's what we're having to fight against.
Do you get discouraged when you mentioned that you don't work with people, seeing an end result of this insurmountable problem, times where you say, man, let's throw in the towel, and I'm being facetious,
right.
But working in such an environment all the time that's kind of away from the end result, the cure.
I think the thing that makes it easy for me to not get discouraged is to be in a community like Austin, because I get to interact with people in the community in an official capacity. I get to go talk to groups, talk to school groups. We have people come in. When we have fundraisers, we get to be involved with that. I get to see how these diseases have impacted people's lives. They certainly have impacted my life. My mother passed away from advanced stage ovarian cancer about 2 weeks before we moved to Austin. So, I mean, I've had a direct impact on my life. So I know exactly the pain and suffering that people go through. I've gone through it. To see people, to be involved with people who are promoting our research, who are giving their time, who are giving their money, and who are giving their support, that keeps me from being discouraged. I don't have to be able to see patients. I can see families who are looking to us. And I'm sort of at the very bottom of the pyramid in terms of discoveries. We're interested in some very basic things that go on in cells. That gets used up the pyramid before it gets anywhere close to patients.
Go a little bit more on that. Where does
Sure.
your research end up in that pyramid? Where does it go?
So we're maybe close to the bottom in terms of basic discoveries. We have work that's directed towards particular diseases, but mostly it's globally on how our cells work. That's going to be used by other people who are going to identify what are known as targets, a particular molecule, a biomolecule in cells that's a particular target for a drug. Then you're going to have another group that's going to actually develop that as a compound, and then decide, is that compound, can you give it to a patient and it doesn't cause them terrible harm? Then it becomes a drug, and then how well does that drug work in preventing that disease or causing that disease to go into regression or curing it. But you can't have the drug part without having the very basic biology. That's really important. So even though we're far away from patients, we also recognize that without this, there's not going to be the development of these new types of drugs.
And not to get, I don't want to get too in the bushes, legalities and all that stuff, but that's got to be a whole other game because, you know, the Institute is part the University of Minnesota, it's part the Mayo Clinic, you have many partners and many other funders. So when someone cracks that code or has that drug that goes to market, you know, we have to make sure that everybody gets credit for it, right?
Oh, there's mechanisms involved. The university has fantastic mechanisms involved for making sure that the researchers and the university get credit for those types of discoveries. For my lab, we really don't have to worry about that too much. We're not a really commercialization-based type of lab, but there are other labs where definitely.
Well, I'm sorry to interrupt, but I mean future funding for your research work, not like, I mean, I understand, I didn't mean commercial like at the magic drug, but you know, there's a problem or an issue at least to talk about maintaining funding and growing funding.
Absolutely.
And to make sure that people out there understand that what you do helps.
Yes. So the funding, most of the funding that runs our lab and most biomedical research labs in the country comes from the public. It comes from the National Institutes of Health, which we apply to grants from them, and they give us dollars to do these research. Most of this research, or most of those dollars are spent on researchers. It's the salaries for people who work in our laboratory. We can't really commercialize anything that we do, so we have to be dependent on those public dollars to do this type of research.
And it's been going down, down, down.
Unfortunately, we had a huge doubling in between the Clinton and Bush administration. So both the Democrats and Republicans worked very well together to put together this doubling of funding, and then we've had a stall. And so we've seen the real ability to do research has shrunk over the last 10 years.
So what can people do, somebody watching, and I drive past the Institute all the time and I just feel it's wonderful, but I don't know anything about science. I know you do good things, so there's nothing really I can do. What can they do to help?
So it's a political issue. It's a political issue for people to talk to their representatives and talk to their senators and say that that's something that they support. It has support in all 50 states. It has support in all political parties across the spectrum. When you talk to legislators, they'll tell you that biomedical research is one thing that everybody agrees on. But it is public spending, and public spending requires the will for people to say, yes, we want to do this. So supporting politicians and saying this is something that we really believe in, this is public spending that we think is important, that's what people can do. Because that's really where we get the vast majority of the support that we have to be able to run these labs.
Because you're fighting a battle even with inflation.
Absolutely.
Keeping up with inflation for your staff.
These are high-tech jobs. These are people who largely have PhDs, so they have options. You know, this is something where there's going to be wage inflation. There always will be when you're talking about high-tech, high-education jobs. So we have to be able to retain people. We have to have some kind of continuity so that we're not constantly starting and stopping. And that's where we've been for about the last 5 years, where you'll get a grant, you'll be able to do some work, and then it's very difficult to continue on with that work. And so that starting and stopping makes us very inefficient.
So something as simple as even going beyond the Institute and saying to somebody, I support the National Institutes of Health, the NIH Funding, or thank you, Member of Congress.
Absolutely.
That would help.
Yes.
That would be something we can do to help.
Yes, and members of Congress and senators, it's something that they want to support almost universally. When I've talked to people from NIH, when I've talked to people from the National Science Foundation, they've never have a problem when they talk to legislators. Legislators love supporting the NIH, they love supporting the NSF, they know the work that we do is good. Getting that support from citizens and voters to tell them we support it too, that makes all the difference.
Well, it's certainly been informative for me, and I'm sure for everybody out there watching. to learn how important your work does and is. And thank you. Congratulations on your success and work, and thanks for your service.
Oh, it's my pleasure. It's a real joy, and thanks for having me on.
You bet. Dr. Ted Hinchcliffe is Associate Professor, Researcher at the Hormel Institute. You can go to their website or ours for more information. We'll have that link on our homepage. For now, I'm Eric Olson. Thank you for joining us. So long.
On Cue is a production of KSMQ Television in Austin, nonprofit public television.